So here we will be discussing regarding sequence alignment. Sequence alignment, you have some idea because we have been doing this during our practicals like BLAST or NCB. In the use of NCBA GenBank also we have collected some sequence. So it's basically sequence alignment. It's a fundamental technique in bioinformatics used for identifying regions of similarity. I will show you one picture here. So this is a sequence alignment. So here the software will be aligning one sequence here, another sequence here, and they are finding regions of similarity. See, you can see the T here, adenosine here, adenosine here, cytosine here, cytosine here. So such a kind of similarity between two DNA sequence will be analyzed in a sequence alignment process. So that is mainly what sequence alignment will be doing. So by doing this sequence alignment, we will be able to understand the evolutionary relationship. For example, here you can see that there are two sequences, and this first sequence and second sequence. One is target sequence, other is Cori sequence. Now we are trying to compare them, and we find that there is a strong similarity between, or nearly 75% similarity between first sequence and second sequence. So that kind of understanding is what we obtain from sequence alignment. So we will be getting whether two sequence or two species are evolutionarily related with each other. So that will be the idea that we are getting from sequence alignment. And here there are mainly two types of alignment. One is pairwise alignment, other is the global alignment and as well as local alignment. Now in the case of pairwise alignment, we take two pairs. This is what is shown here, the first one. There is a pairwise alignment. This is one sequence, this is another sequence. One pair of sequence will be compared with each other. So pairwise alignment involves comparing two sequences. So if you hear the term pair, there will be only two sequences. So two sequences will be compared with each other, and we are trying to find out the best possible match between them. So this is how we are trying to compare two sequences. One is a first target sequence, another is a Cori sequence. So by doing this, we will be able to identify the matches between them, best possible matches between them. And it is mainly used to determine the degree of similarity, how much similar these two sequences are, and also to find out regions of conserved region. Conserved regions means those regions of DNA where there is little change during evolution. Here, for example, this is these two regions are generally known as conserved region. That is, during the evolutionary process, they will not be there won't be any changes happening in those regions. So such a region which are evolutionally more or less permanent are known as conserved region. There is no evolutionary change taking place in them. So that is the idea behind the pairwise alignment. We are comparing two sequences. Now if you take the pairwise alignment, we can classify that into global alignment as well as local alignment. The picture that you are seeing here is one is local alignment, another one is a global alignment. So in the case of local alignment, we are focusing on finding most similar sequence within the two larger sequence. It is especially useful when sequence vary significantly in length or share only partial similarity. For example, here you can see that this is one sequence, this is a second sequence. We will be trying to align those regions where there is maximum similarity. So if we put the sequence like this, we can find that in this region, in the middle of the sequence, we can find maximum similarity. Whereas the second sequence, what we are trying to do is that this is the first sequence and this is the second sequence. We try to spread it from starting point to ending point. So here you can see this same sequence, we are trying to stretch from this end to 3' end. So he can see that only in certain regions we are finding similarity. For example, up to this there is similarity. Then there are 4 gaps are here, and during that gap there we cannot find any regions of similarity. So we put a gap or dash there. So, but advantage of global alignment is that entire sequence is aligned. So align 2 sequence along their entire length, optimizing the matches across the entirely of both sequence. So what we are trying to do here is that we are trying to spread out the sequence, and we are trying to find out where there is maximum similarity. We will be aligning them. Where there is no similarity, we will be putting a dash or gap there. So that idea of spreading across the whole sequence is known by the name global alignment, and other one is a local alignment, where we will be aligning only on regions of maximum similarity, is known as the local alignment. So this is an idea between what is a local alignment and what is a global alignment. Then another type of alignment is known by the name multiple sequence alignment. Here you can see that we are only using 2 sequence, but lower section you see that here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 sequences are aligned with each other. When more than 2 sequences are aligned, we call it as multiple sequence alignment. So in the case of multiple sequence alignment, we can find more than 2 pair of DNA sequence. So that is the idea of the multiple sequence alignment. It involves aligning more than 2 sequences simultaneously, means at the same time, to uncover conserved regions or across a group of sequence. In that different type of sequence, which are the regions where there is maximum similarity, which are the most permanent or most conserved region in that sequence, in that group of sequence. So that is the aim of this multiple sequence alignment. Now here you can see that again the global alignment aligns the entire length of both sequence, ensuring that end-to-end alignment is optimized. This approach is most effective for sequence of similar length and closely related species of protein. For example, here you can see that this is the one sequence and another sequence. We were able to align like this because the both sequences are of somewhat same length. But if there is so much difference between the first and second sequence, global alignment may be impossible. So only if the 2 sequences are of similar length, global alignment is possible. Otherwise, it may not be possible. The Needle-Wunschmann algorithm is commonly used for global. It's a software technique, and that is the method by which we are aligning the global alignment. And in the use of this programming, they will be using dynamic programming to calculate the optimum alignment by maximizing the score of matches while minimizing the mismatches and gaps. So it's a software technique, and that software is known as a Needle-Wunschmann algorithm. Then another is in the use of local alignment, identify the most similar sequence between 2 sequences, disregarding flanking region that do not align well. So flanking means bordering regions. So here you can see that this is the local alignment. We are not bothered about these 2 portions. We are simply skipping that region or disregarding that region. We are only matching or aligning where there is maximum similarity. So that is the feature of a local alignment, and this is mainly beneficial for comparing distantly related sequence. That's one advantage of local alignment, or finding conserved region within a long species. So when there are 2 separate sequences and their evolutionary relationship is very much wider, then we will be using this method. Now here the algorithm that we are using is Smith-Waterman algorithm. So that is the algorithm that is used, or software technique that we are using. It focuses on high scoring subsequence while ignoring poorly matching regions. Now here the how this alignment is actually done is basically based on giving some score. Sometimes positive scores are given for matches, negative scores are given for mismatches. There is a technique for that. I am not explaining that. And based on this, there are different types of metrics like PAM and BLOSSOM are two commonly used alignment technique to align similarity scores based on evolutionary consideration. So this is a method used for finding out the how regions of similarity are present in two sequence. Now coming to the application, it's mainly identifying homologous genes. You have studied what is a homologous genes in your genetics. Then it's another advantage is that finding the evolutionary relationship between two sequence. Then annotating genomes and functional, that is given the function, what is the function or given explanation for certain genes, and dictating mutation and polymorphism. So by this kind of method, we will be able to find out regions of similarity where there is difference, where there is mutation, where there is polymorphism. All this can be explained based on this simple technique of alignment.